Okay, if you want to go ahead and get started, Jack, whenever you're ready. The timer function does not appear to be working in the breakout room, so we're going to ride the
Okay.
waves and just know that you've got 10 minutes. You can go when you're ready.
All right. I got one on my phone, so I'll be okay. Well, thank you everybody for being here. Um, I'm a first semester graduate student, so I don't have any results on this yet. This is just my proposed research. I'll be doing my proposal for it next semester. But I'm going to be studying steelhead trout in Lake Erie. Um, the steelhead trout fishery in Lake Erie makes millions of dollars for the state, just based off like tackle and bait sales and the whole industry around the angling. And there's recently been some changes in management, namely by us and by Pennsylvania, that are causing some concerns. Um, but there was a study done in 2018 by Budnick et al., where he found that the majority of the fish that they're catching in Lake Erie tributaries in New York are being stocked from Pennsylvania. Um, Basically, this implies that our fishery in Lake Erie is heavily dependent on the stocking practices in Pennsylvania. Um, and in the past year, I think 2024, they reduced by 20% the amount of steelhead they're stocking. And then this year in 2025, they added an additional 50% reduction of steelhead being stocked. So the New York tributaries are expecting around 2027, when those fish that they stocked starting to spawn, that we're going to start seeing less and less returns. So my research is basically figuring out where are all the different fish in the Lake Erie tributaries coming from, so we can establish a baseline right now um, about proportional how many fish are being stocked in Pennsylvania, how many fish are being stocked in New York, how many fish are from Ohio and Ontario. So it's figure one right here. I have my three sample sites. We chose these three sites because they're all management priorities. Canada Way, Cattaraugus, and Chautauqua Creeks near Dunkirk, New York, which is also pretty close to the Lake Erie Fisheries Unit for DEC that I'm working with, so that's very convenient. And Cattaraugus and Chautauqua Creek both have potential for natural reproduction as well. Um, and they receive a lot of fishing pressure. Just so these three creeks make a great jump off point. Cattaraugus and Chautauqua were also both included in that previous Budnick study, so we have data on them from 2018 about the proportional makeup of where all the fish are coming from. So the questions that I'm actually going to be asking are, what are the relative proportions of stocked and wild? So two of those creeks that have potential for natural reproduction, is that actually occurring there? And just overall, how many fish that are returning are actually stocked, not looking necessarily at where they're being stocked from. And then my second question is, where are the stocked fish from originally? So that's actually figuring out, okay, are they from Pennsylvania, Ohio, New York? There's only 7 or 8 tribute or hatcheries that stock these waters in New York or Lake Erie. So we can use something called otolith microchemistry to figure out basically where they came from, and I'll talk about that in a little bit. Um, we're going to be figuring out the rest is kind of just, it's a very good opportunity while we're sampling all these fish to provide information on the fishery for managers, because it hasn't been evaluated in a long time. So we'll be looking at the average age of the steelhead trout population returning, the condition. So it's basically, we're going to be measuring Fulton's K. So it's like a length over weight, how healthy are the fish metric. And interestingly, I want to see if the wild fish are doing better in the wild than the stocked fish are. So are they more fat? Are they eating better? And then we're also going to be categorizing the diet of these steelhead trout and seeing if there's any difference in what they're eating between wild and stocked fish. Uh, oh, for aging too, figure two up here. You can see, pretty cool. It's kind of like a tree. This is called an otolith. It's an earstone it's formed abiotically. So it's just influenced by the environment, not the fish's biology. And deposition rates change as the temperature changes and with seasons. So you get these rings on there called annuli, and you can measure them, or not measure them, you can count them to figure out how old the fish is. So there's the center, and then one ring would be 1 year, and then two rings would be 2 years, and so on. Uh, so we're going to be collecting these from adult fish that we're obtaining from the mouths of our 3 locations by either boat or backpack electrofishing. So Cattaraugus is too deep to wade in, so we're going to be using a boat electrofisher there. And then the other 2 are going to be backpack electrofishing. So we're going to take general biological measurements, length, weight, sex, if it's possible to observe that. Sometimes it's not. And then multiple different tissue samples for our analyses. We're going to be attaining strontium-86 and barium-138 ratios using laser ablation inductively coupled plasma mass spectrometry. I know that's a mouthful. I'm still learning about that, but it seems pretty cool. It's basically just looking at these different element ratios in comparison to the ratio that they occur in naturally in the environment. And since these elements are deposited abiotically, you can basically get a signature of each location based on these certain ratios. So Salmon River Fish Hatchery in New York will have its own signature. Fairview in Ohio will have its own signature, and so on. So we can actually create something called a linear discriminant analysis, where we can take an otolith out of an adult fish and measure these isotopes in it, and then figure out which hatchery it came from. So we'll be collecting yearling fish from all these different hatcheries that stock Lake Erie. I don't know how many we're going to be collecting yet, kind of at the beginning of my project here, but we're going to be using that to classify where the adult fish that we catch come from. And then diet trends, we're going to use fatty acid signatures and stable isotope analyses. Fatty acid signatures are basically a reflection of what something is eaten in the past month or two. Fatty acids are transferred from prey to predator fish conservatively. So if a predator fish eats a prey fish, that prey fish's fatty acids are going to be retained in the tissues of the predator fish. And you can measure that and figure out what the predator fish has eaten based on those fatty acids in its tissues. And then stable isotope analyses, kind of similar to the otolith analysis, where we're just going to be measuring different isotopes that have to do with trophic level and primary energy. So carbon can be used to track basically like what trophic level that fish is eating at, and same with nitrogen. And we're going to be doing that using an elemental analyzer. I don't know where yet, but the fatty acid analysis we do in-house here at SUNY Brockport in our lab with my advisor. Okay, I talked about everything. Oops. Figure 3 down here, that's just a nice picture of what the backpack electrofishing is going to be like. That's Jared from, he's a biologist from DEC, Lake Erie Fisheries Unit. And yeah, that's going to be my project. Sorry, I don't have any results to talk about, but
Okay, well, we don't really have time for questions today, so you've still got 1:30 left. If there's anything else you want to share, if you want to talk about where your project's going next or anything. In the meantime, Caleb, if you'll be aware you're on deck next.
yeah, this summer we're going to be doing some sample processing in the lab. Here we're going to probably be doing fatty acids, and I'll be preparing my otoliths. So we have to make them very, very clean. We can't use metal instruments on them because of the nature of what we're doing. You can't get any extra isotopes in there. So that's going to be a long process, and I haven't done it yet, but I'm really looking forward to it. Yeah, that's about it. Thank you guys for coming. I appreciate it.
All right, well, thank you for presenting, Jack.
Mhm.
I'm going to go ahead and unpin you, and we'll give Caleb a minute. Caleb, when you are ready, let me know, and I will start a 10-minute timer for you. Again, that's not meant to be any pressure. It's just to help you pace yourself.
Great. Yeah, thank you. Let me start to share my screen here.
We can see your screen, but you probably want to focus on the slide itself, and your 10 minutes is going whenever you're ready.
Gotcha. All right, how's that look? You guys able to see my poster here?
You're good.
Great. Thank you very much. Yeah, so thanks everyone for being here. Happy conference day and a good afternoon to you all. I will be presenting a poster that I've put together about my proposed research, as well as some preliminary results from my first sampling season. So I'm a graduate student here at Brockport. I started in the last fall, and was able to sample some lake trout as well as some prey fish. So I do have some preliminary results that I'll get to towards the end. But I will start with just a brief overview of my species of interest, that being the Great Lakes lake trout. They are a native fish and a deep water piscivore, meaning they are fish-eating predators, and they tend to occupy the apex predator role in many Great Lakes food webs. They play a lot of different important aspects in the lakes. They're ecologically important because they control the food webs through top-down control, keeping primary consumer populations in check and balanced through predation. They are commercially important as well to us as humans, offering opportunities for commercial fishing as well as recreational fishing and building economies in that sense. They also play an important cultural role in the Great Lakes region. They have been harvested for many centuries here in the Great Lakes, and the lake trout was once ubiquitous across all 5 Great Lakes in great numbers, but somewhere right around 1950, we saw a pretty devastating crash in lake trout populations across all 5 lakes. And this has been attributed to overfishing as well as sea lamprey predation that caused the lake trout populations in Lakes Michigan, Erie, and Ontario to be nearly extirpated in the mid-1950s. So to combat this, there has been some stocking initiatives put in place through the Great Lakes Fishery Commission. And by 1978, all 5 of the lakes were being stocked with lake trout to help boost their populations. And this has been successful in many of the lakes. Lake Superior just recently in the fall of 2024 announced that their lake trout fishery is fully recovered and consists of self-sustaining populations. So that's good news up in Lake Superior. That is the ultimate goal for these fisheries, is to have self-sustaining populations. But in Lake Ontario, a little closer to home here, the lake trout populations are still struggling to recover, even with stocking influence. So we want to understand what might be preventing these lake trout from regaining their population abundance. And we propose that it could be this method of thiamine deficiency that's causing early life stage mortality of their offspring. So I'll start by saying that thiamine is a vitamin B1, that's essential for metabolic processes and energy production in all vertebrate life, not just fish, but all vertebrates require this vitamin. And vertebrates cannot synthesize thiamine, so they must take it up through their diet, consuming thiamine-containing organisms like bacteria or phytoplankton that are capable of synthesizing vitamin B1. We propose that the method of thiamine deficiency is through diet composition, and there's two hypotheses that seek to link diet composition to thiamine deficiency complex. The first being thiaminase. So thiaminase is an enzyme that's capable of breaking down thiamine in the gut of a predator. So if a lake trout is consuming prey that is high in thiaminase activity, it's likely that their thiamine levels will be decreased through that process. The second hypothesis is that excessive unsaturated lipids can lead to thiamine deficiency if it is using up thiamine as an antioxidant. And that's through this process of lipid peroxidation where the unsaturated lipids are being oxidized in the fish's tissues. So some objectives for my research, I seek to compare the nutritional quality of prey species from Lakes Ontario and Lake Superior to better understand their thiamine concentrations, lipid content, fatty acid signatures, as well as thiaminase activity. And then investigating this relationship between their diet composition and thiamine concentrations. So to understand lake trout diet, we'll be using stomach content analysis, stable isotope analysis, and fatty acid analysis to get an estimate of lake trout diet in these lakes. Our lake trout are collected in the fall of each year during their spawning season. That will be done through gillnet surveys by multiple agencies, including us here at SUNY Brockport. So I have a map, figure 2 on the bottom of my sample sites. The 3 sample sites in Lake Ontario include Olcott, Hamlin, which we'll be sampling at SUNY Brockport, and Oswego. And then in Lake Superior, our collaborators will be running gillnet surveys at the Apostle Islands, in Keweenaw Bay, as well as in Munising. And when we capture these lake trout, we'll be taking some tissues from them, including the belly flap, muscle plug, the eggs, stomachs, as well as a maxillary bone that can be used for aging. The belly flap will be processed to understand the lipid content as well as fatty acid analyses. We'll use the muscle plugs to measure stable isotope ratios in each individual fish. And we'll be using the eggs for thiamine concentration. We'll also be collecting prey for this research, and that will be done via bottom trawl surveys at multiple different locations throughout the lakes. And the prey fish that we're targeting are shown here in Table 1, right in the center of the poster. From Lake Ontario, we'll be collecting alewife, round goby, deepwater sculpin, rainbow smelt, and mysis. And the latter two actually overlap with Lake Superior, where we'll be collecting rainbow smelt, mysis, as well as cisco, bloater, and nine-spine stickleback. And we'll be analyzing the thiamine concentrations of our prey, as well as the thiaminase activity, if any, and some fatty acid analyses as well for our prey. Quickly, with my last minute here, I'll just go through some of these preliminary results of thiamine concentrations from our fall 2024 lake trout samples. So this is from 74 female lake trout eggs across 4 sites in both lakes. And you can see in this box plot here, we found significant differences in total thiamine concentrations of lake trout eggs between Lake Ontario and Lake Superior, with Lake Superior concentrations being significantly higher at both locations compared to the ones found in Lake Ontario. This dashed red line here shows the thiamine concentration threshold for thiamine deficient or sufficient eggs. and that is set at 4 nanomole per gram. So the thought here is if the eggs contain greater than 4 nanomole per gram of total thiamine, it's likely that they are healthy and will have not experience early mortality of the offspring and a good survival rate. But if they fall below this 4 nanomole per gram threshold, it's likely that they will be thiamine deficient and experience poor survival of their offspring. Thank you all for your time. I appreciate you listening and hope you have a great rest of your conference.
Thank you for that presentation, Caleb. We're going to give just a minute. I have 2 instances of Amina, so I'm going to pin both because I'm not sure which is the correct one. And Mina, you're going to be up next. When you are ready, let me know and I will start the 10-minute timer.
Hello, can you hear me?
I can hear you.
Actually, I have to join from 2 different places because of my something wrong with the mic earlier.
Okay.
So yeah, I'm just gonna share my screen to everyone. Can everyone see it?
It is showing now. I'm going to start your 10-minute timer, and I admire your creativity and problem solving. So well done.
Thank you so much. So hello everyone. First of all, welcome. And today I will be introducing you all to one of my. I'm a 3rd-year PhD candidate, and I'm introducing my research project that I'm actually doing right now. So which is based on the biogeography of sediment and fish gut microbiomes in the Great Lakes. So as we all know already, the Great Lakes over here, we have these Great Lakes. They are like 5 in number. And essentially, these lakes are further divided into 2 different categories, which are one of them is more contaminated and one of them is less contaminated area in the Great Lakes. So I'm focusing on that. So what we are doing in this study is to investigate the microbiomes of the lake trout and also the sediment microbiome in these Great Lakes from both of these areas. And what we are trying to do is to see how these mobile genetic elements found in these microbiomes are basically driving the ecology of the microbial ecology and also the evolution, and also how these different emerging contaminants that are actually found in these Great Lakes are affecting overall microbiome in the less and the more contaminated areas in the Great Lakes. So our hypothesis includes like the environmental conditions and contaminant profiles basically shape the sediment microbiome. And since we are focusing on the lake trout, which is the top predator in these lakes, that is actually being influenced by sediment, fish physiology, and also the contaminants in the environment. So we are trying to see how these microbiomes can reflect the overall health of the ecosystem and especially of that of these Great Lakes. So starting with I have some of my preliminary results over here too. So starting with our project design and method, so we do get like fish fecal material and also the sediment samples from the EPA. And then we further process them. We have some information which includes fish age, weight, and gender. Further, we also like trying to see how different contaminant profiles are present in these lakes based on the information given by the EPA. So we are doing some microbiome analysis and seeing the relative abundance of the different antibiotic resistant genes in these lakes, and further how these microbiome structures are, what are the composition and structures, and also the functioning of the microbiome by using different methods. So we have done a lot of DNA extraction of the fish fecal material and also the sediments, and then we further do the DNA quantification and fragmentation analysis using different methods, including qubit, Agilent, biotech, everything. From there, we further do the PCR and qPCR. PCR is basically just for the to see if there are like antibiotic resistant genes that are actually present or fecal biomarkers that can be actually present in these samples in order to see how these mobile genetic elements are working there. And then we are already done with the 16S amplicon sequencing So basically, we are targeting 4 lakes, Lake Huron, Erie, Ontario, and Superior. As of now, we don't have samples from the Michigan yet. So we are all done with the 16S amplicon sequencing, and further we are moving towards the shotgun metagenomic sequencing, which we will be doing soon this summer, hopefully, which includes the Illumina sequencing and also the NanoPro sequencing. We are trying to join both of them together. So for our results, I have just put in some basic map of the Great Lakes and to show different areas. All the ones that are in boxed are the ones that are industrial areas, so more contaminated areas, and the ones that are not um, contaminated are the one without the boxes actually. And also these different arrows shows the lakes that we are working on these days. So I have this small heat map showing the presence of different antibiotic resistant genes. So we actually did up to 24 different types of fish of the antibiotic resistant genes, mobile genetic elements, and fecal pollution biomarkers for the Lake Erie and Superior as of now, both fish fecal samples on fish fecal and the sediment samples. And we did find like there are very sediment has more of these mobile genetic elements as compared to that of the fish fecal samples. And from our 16S analysis, we have these amplicon sequence variants, which are the ASV counts, in order to see the presence in the fish fecal samples and the sediment samples. And we did find the fish fecal samples have relatively low number of ASVs as compared to that of the sediment samples, which is quite understandable because the sediments are like way more complex as compared to that of the fish. Further, from our 16S analysis, we have seen the PCOA plots for both of the sediments microbiome and the fish fecal microbiome is over here. From sediments, we have seen that all the lakes, all of them are like pretty segregated, and all the samples from the same lake are like together, but they're far off from each other, showing that they have like different microbial sediment composition. But if we look at the fish fecal samples, it's quite interesting because most of the fish fecal samples are quite integrated together, and there is a lot of overlap between the lakes which are close together. So if you look at Lake Erie and Superior, Erie and Ontario here, which are closer here, so the fish is like closer over here, and for the Superior and Huron, which are like technically a little bit closer to each other, also have a lot of overlapping over here. So for our results, I can say that the sediment has higher presence of antibiotic resistant gene, mobile genetic elements, or fecal pollution biomarkers in the fish fecal samples. And then we have higher ASV counts, which is also then understandable because sediment is more complex as compared to the fish fecal samples. And sediment microbiomes are more segregated as compared to that of the fish fecal samples. So we are trying to, so far we have these preliminary results, um, and we are further interested in completing as the samples are coming in from different lakes. We're trying to get more of the information regarding the antibiotic resistant genes, and further we are also at the same time doing a metadata analysis, all the existing studies that are actually present, and using that information we also want to relate that information to the current microbiome composition in order to see like how the historical trends of the contaminants is shaping the overall microbiome. So I would like to acknowledge a lot of people, including the SUNY Empire Innovation Program and also the EPA and Clarkson, who we are collaborating with. So yeah, that's pretty much it. And thank you, everyone.
Thank you for your presentation. You do still have another minute or two if you want to talk about maybe where you're going next with the project.
So these days we are highly interested. As I mentioned already, so we have shotgun metagenomic sequencing lined up. So we are interested in seeing the overall DNA structure or DNA sequence in order to better understand what is the composition, what is the structural composition of these microbiomes. For that, we do plan on using the nanopore and also the at the same time Illumina sequencing. Both of them are like different because of one will give us the short reads, the other one will give us the long reads, and we want to actually overlap in order to produce the de novo sequencing. So we are really interested in that. We are actually working on that these days, and hopefully we will be done by the end of summer with um, this part of the project too. So yeah, it's pretty exciting. I think it will definitely help us to understand better uh, how um, how the microbiome that is very significant, but we don't really think about it that much. In the Great Lakes can be overall shaping the overall ecosystem of the Great Lakes. So um, I find it very interesting and exciting all the time, and results are always very um, fun to explore. So that's what I would like to say as of now. So I hope other people can enjoy our research as it gets published and everything.
Fantastic. Thank you so much.
Okay, and stop sharing if someone.
I do not see our next presenter, Sophie Neubauer, in the room, nor co-presenters Cole Ross or Chris Fernandez. So missing that group, Darian, are you ready to go next?
Yep, I can go.
Okay, whenever you're ready, I will go ahead and get your timer started.
Okay.
When it unpins Amina, hang on. Here we go.
Um,
Are you also sharing your screen?
I'm attempting to, but
Technology is wonderful until it isn't. Hang in there, you've got a few extra minutes because you're going early, so no pressure.
okay. I think it's because I just re-downloaded Zoom. It doesn't have the permissions.
Okay. You should be able.
Okay, if I quit Zoom and come back, because it's
Yeah,
not.
you've got time. Why don't you jump out and come back? We'll wait
Okay,
for you.
thank you.
While we wait for Darian to jump out and jump back in to see if they can share their poster, I wanted to let people know the recordings for these poster sessions will be posted within the next few days. I've put the link in the chat. And if any of our presenters who were not able to go at their original assigned time join us later, I'll just catch up to anyone who is here at that point. So we're just waiting for Darian to get a chance to log out and log back in and see if they're able to share their screen at that point. It looks like from the list of presenters I have, Jonathan, you might be next on deck, although you weren't originally scheduled to go till later. If you want to kind of get yourself ready, it looks like you will be next on deck.
Sounds good. Thank you.
Thank you.
Actually, there's a couple of us here, myself and Kate Henderson, who are in Tanya Plumey's class. So she registered, but we are here to present.
Okay, fantastic. When we get
That's
that name, just shout out and let me know. Looks
yeah,
like Darian is logging back in, so we will give them a minute.
cool.
Okay, we can see your screen. So if you'll tell me when you're ready, I can get your 10 minutes started.
Okay,
Again, no rush. Take your time.
let's see. Is it showing the presenter view or the actual poster?
It's currently showing presenter view.
Not now.
There you go.
Okay. I'm ready to start my time.
Take it away.
Okay. Oops. So I'll be presenting about my project looking at the hybridization and population structure of water buffalo species in Lake Champlain Basin. I am from SUNY Oneonta, and this is a big collaboration project. I am a first-year master's student. There's another student working on this project. My advisor, Alex Atola, and me are working on the genetics side, where Kaylee and Dan Stitch are more on the statistics side and looking at diversity. But I will be talking about the genetic side of this and the survey of milfoil in Lake Champlain Basin. So what we want to do is we want to be able to get out there and do these plant surveys. And as we're doing those, I will be collecting water milfoil. All of those samples, we want to be able to extract the DNA from those. And we want to do that so we can have a population genetic survey and also have a genome-wide association study. So that's essentially kind of, if we're looking at lots of different lakes, we want to see, are there genetic differences between those lakes? And within a lake, are there genetic differences between those individuals? And the genome-wide association study is kind of looking at different phenotypic traits and seeing, are there genetic markers that can help us better understand like why we're seeing different morphology in different plants, even within the same species. And we want to have that, so if there are something that is useful for identification, we can update identification guides for milfoils. And we also want to make sure to publish the survey data so that's available to lake management associations. And hopefully the goal of this project is to help lake management associations make more informed decisions considering water milfoil. And that is because one of the main species in this project, Eurasian water milfoil, is an invasive species. So it is a pretty large concern, especially in the New York and Vermont area where this study is taking place. So last year we were able to get out to 8 New York and 6 Vermont water bodies, and we sampled for water milfoil tissue. And this summer we're hoping to get back out and hit at least 15 additional lakes, including Lake Champlain as well as Lake George. So if you look at the species of interest, these are the species that we came across. So we have our northern, variable, Eurasian, and then we also have a hybrid species. So the northern is the native species. We actually haven't seen a lot of it. The variable is also considered invasive, and then the Eurasian is the main focus of the study because this is the invasive species that people spend a lot of money every year like trying to manage this. And it's able to hybridize with the native species, creating a hybrid. And the reason this is important is because those hybrids show these intermediate traits and they're very hard to distinguish. So you need that genetic component to a study to truly be able to know like what individual you're looking at. And the hybrids also, on average, will show more aggressive growth relative to the parental species. And they sometimes even show herbicide resistance. So it's a concern for management. And that's why we want that data to be given to them so they can treat their populations in the best, like most effective way. So I do have some preliminary phenotypic data. So the PCA that I have, this is just based on like what I thought the plants were morphologically. This isn't including any genetic data yet. But the most important takeaway from this is you can kind of see there's a big overlap with Eurasian water milfoil over like northern. And what that is kind of telling me is that within like at least the continuous traits, so like stem length, length in between nodes, which is kind of like the part in between the leaflets whorls that you can see in the pictures. Things like leaflet pairs and the size of the leaves. There's a lot of overlap for those. And that's kind of indicative of like how invasive species are because they typically show way more phenotypic plasticity relative to like their native counterparts. So that could be going on. There could also be hybrids hidden within this data that we might uncover later that could explain some of that variation. And that's also kind of telling me that some of these traits that are often used, especially leaflet pairs, is often like in keys for IDing these species out in the fields. Those might not be super reliable, reliable, and that has been like kind of confirmed already in literature, but this would just be kind of something on top of that. Like, again, like you really do need to do the genetic work to know confidently like what species you're looking at. So the next steps, we're currently working on processing all the genetic data. That's kind of been the main task for this semester. and we're going to want to try to determine where our hybrids are and all the true species identifications. We want to try to get at the genetic structure and also the hybridization dynamics. So with hybrids, sometimes it could be like unidirectional or like kind of one species is often like maternal or paternal. So we want to recognize any kind of patterns like that. Again, we want to look for associations between the selective phenotypes and genetic markers. And we're wanting to return to Lake Champlain Basin area this summer for more intensive genetic sampling. The only main changes this year is that we will be collecting more Pacific data. So it'll be a similar amount of samples, but we're going to be taking down like longitudes and latitudes. So that way we can create maps. And once we have our genetic data, we would be able to have maps and identify like where the different species lied or where hybrids lied. So if there was a patch of hybrids within a lake, maybe that could be treated separately than the rest. And the lakes will be selected based on predictions of occupancy detection and species richness from year one data. And that's the other part of the project, which is Kaylee's part, which is understanding diversity. So that's more on her side. And I also have my works cited here. And for acknowledgements, I would like to thank NUEPIC as well as the Lake Champlain Basin Program for funding this project. I would also like to thank my advisor, Dr. Alex Atola, for all of his support. And that's about all I have for this.
Okay, did you want to say anything about where you're going next with your last 2.5 minutes, or do you want to turn it over?
I can turn it over. I think I said most of what we were planning to do next already.
Okay, then let's restart the timer. Let's look at, do I have anyone from Sophie Neubauer's presentation in the room? Okay. How about Konstantina Plumey?
Yeah, that's myself and Kate who's here.
Okay, so that's Rob and Kate. Let me get Rob and Kate pinned. Okay. Do you need a minute to prepare, or do you want me to go ahead and get you started with your timer?
I think we're good. Rob, do you want to share the poster or do you want me to share my screen?
I can share mine, and we can. I didn't know who else was going to be on, so we can kind of tag team whatever information's there. So let me go ahead and share my screen.
I'm going to start your 10-minute timer. Thank you for your flexibility on your timing. Take it away. Okay, we can see your poster.
And Rob, I think you might still be muted.
Oh, yes, I was. Sorry about that. Can everybody see the poster still?
Yes, we can.
All right, great. So I'm Rob Katz. With me on the call is Kate Henderson, and we're two students from Dr. Tanya Plumey's class, along with Karen Lindberg's and Stacy McNulty. We're from SUNY ESF, and we developed this research for a course that is focused on evaluating the ecosystem services of SUNY ESF's regional campuses. So SUNY ESF has a number of different satellite campuses that encompass a really large area of land in New York State. And these encompass a different variety of habitats and uses for those habitats. So there's over 25,000 acres of regional campuses beyond just the SUNY ESF campus in Syracuse. And what we were addressing in this course was, how do we look at those campuses and assess and evaluate the ecosystem services that they provide? So that term ecosystem services are the benefits that people derive from nature. These can include things such as, the ability of plants to purify the air, the ability of wetlands to purify water and help with stormwater retention. But they can also include things like provisional ecosystem services, cultural ecosystem services, which are more kind of human-related activities. So, deriving revenue and profit from the natural resources that are on these properties, which does happen, as well as, the general availability of these sites for people to go out and take a hike or bird watch or other activities. So each of us, there were 8 students in the course, I believe, and we divided into several groups which focused on different ranges of ecosystem services. So these were provisioning ecosystem services, biodiversity ecosystem services, cultural ecosystem services, and regulating ecosystem services. And we looked at those across as many of the campuses as we could. What we did find is that some data sets were more thorough than others, depending on the campus. So myself and Todd Waldron, who is a student in the course, we looked at the cultural and provisioning ecosystem services. So we identified 14 distinct areas of cultural ecosystem services, including 7 unique cultural systems. These included things ranging from hiking and birdwatching to the direct sale of maple syrup and lumber from SUNY ESF properties. And these, totaled between the cultural and the provisional and the recreational ecosystem services, we calculated an estimated over $57,000 annually is contributed to the local economy from just a couple of the satellite campuses that SUNY ESF has. And Kate, if you would want to talk about the biodiversity ecosystem services, I can hand it over to you.
Perfect. Yes, I can hop in there. So I was in one of the groups focusing on biodiversity. There was another group doing some kind of GIS and mapping work who incorporated some eBird data here and iNaturalist data as well. So biodiversity, this is really interesting in the realm of ecosystem services. Some people consider biodiversity to be a service itself, whereas other people see it as more underpinning the other services. But regardless, this is very important because it is the species in an ecosystem that are driving the delivery of all these services that are benefiting the people who are also living in that system, using that system, interacting with it. It's also important kind of in the context of climate change, of land use change, because while we know biodiversity is important to all these ecosystem services, there's some disagreement about the exact shape that this relationship takes. Do you have a linear decline in services as biodiversity decreases? Is it resilient up to a point and then really falls off a cliff? Or is it just hard to quantify any kind of direct relationship? So we are interested just in taking the first step of figuring out what diversity do we have across all these regional campuses, which are located in central New York, in the Adirondacks, and the Thousand Islands region of the St. Lawrence Seaway, just to kind of set a baseline for what biodiversity do we have, what do we know about this diversity, and what can we compile. So we'd focused on, for kind of as a pilot study here, a few of these sites with the hope that more work will be done in the future at the Cranberry Lake Biological Station in the Adirondack Mountains, which is a very important undergraduate teaching station for summer courses, as well as a research site. There have been 119 articles published going back to the 1970s. And in those articles, we have mentions of more than 300 species at the field station. However, in a lot of cases, this is biased towards taxon that have had major research studies. For example, we have fungi and terrestrial plants are very well represented here, whereas things like reptiles, amphibians, some aquatic species, and birds are underrepresented compared to what we know is there. So we see there's a lot of really rich information in these published data sets that compiling that will give us a lot of useful information for the future and can kind of identify where those knowledge gaps are. At the Newcomb campus, we've seen really strong research on birds and fish. And then in the Thousand Islands region on the St. Lawrence Seaway, there's been a huge amount of aquatics research. So we see these stations are biodiverse. They are supporting a lot of species underpinning these services. And we have a good start to our data set, but there's definitely a lot of work that can be done with compiling all this information we have, identifying those gaps that have had fewer studies, and directing our research there in the future. We do also see citizen science can play a major contributing role here with this iNaturalist data on species such as birds is a very great complementary data set to the more formal research on what kind of species people are seeing and are mattering to people at these sites.
Yeah. And the main kind of takeaway for all of our groups, I think, was that there are so many opportunities to continue this research in the future. There are opportunities at ESF, which does have a variety of faculty who we spoke to who do study, how do you measure these ecosystem services in a monetary way, or otherwise. And there's still a lot we can learn. There are many opportunities in all of our fields to capture more data and kind of add to that total. But the main takeaway for all of us, I think, was that, looking beyond just the kind of direct financial interaction between people and these campuses, there's a lot going on between the surface that supports the economy that we can value as a service beyond the educational value and beyond beyond how we directly interact with these sites. There are very passive ways that these sites, in effect, generate revenue for all of these campuses. Another big takeaway for all of us, and something we wanted to express as well, was that these are ecosystem services that are not unique to the ESF satellite campuses. They can be applied in a number of different regions, especially across the SUNY system. So if there are I know there are other SUNY campuses with field stations or with any kind of natural area. These are some of the same fields that could be applied to those sites as well. So this is certainly something we were excited to explore, and definitely something that can be applied across the board to other SUNY campuses, and that a lot of other campuses are taking stock of, their not only their environmental impact, but how is the environment that those campuses have contributing to the school itself in an economic way, but also in a more holistic way. So I think that covers a lot of our research. I'll hand it over to Kate again.
Sure. I think my kind of last point that I'd like to end on here is that this field of ecosystem services really came to prominence in the 1990s based on this idea that since a lot of these environmental services don't necessarily have a dollar value automatically attached to it in a clear market, these things were often being undervalued in decision making. This is even to see today as research budgets are tight, educational budgets are tight. Things like some of these regional campuses and these field stations can just be viewed as a major expense. So one goal of this class was really to highlight all of the value that these stations provide in terms of education, in terms of water cycling and temperature regulation in these regions, in terms of the kind of services they're providing to the communities who are taking classes here, hiking on these trails, buying maple syrup at the maple sugar weekend. So just really a good way of demonstrating the broad value of these regional campuses.
Thank you all for your time, and hope you all have a great rest of the conference.
I mean, that was just perfectly timed. Thank you both. That was outstanding. All right. Thank you for sharing, and not seeing the next presenter yet. It may be because they didn't realize the timing has changed a little bit. Jonathan, would you be ready to be up?
Yeah, I can present.
Okay. Whenever you're ready, if you want to share your screen, I will then get your time started.
Right, so hopefully everybody can see this.
We can
if
see
you
it
can't, let
now.
me know. If you can't hear me, let me know.
We can hear you and we can see your poster. So when you're ready, take it away.
Perfect. So I'll be presenting a little bit about some of the research I'm doing here at SUNY Brockport. My focus of my research is on evaluating thiamine deficiency in lake whitefish as a potential cause of the recruitment failure here in the Great Lakes region. If anybody's not familiar with lake whitefish, so lake whitefish are a relatively large benthivorous species of coragonid fishes. They're native to each of the Great Lakes. And in each of the Great Lakes, they play vital ecological roles, especially to the aquatic food web. So as a benthic predator, they're often found migrating between both nearshore and offshore waters, feeding on benthic prey items, and this helps to essentially or greatly contribute to nutrient cycling within both the benthic and pelagic zones of the Great Lakes. Additionally, lake whitefish also hold immense cultural value, especially to the indigenous communities throughout this region, and which have relied on this fish and this species for thousands of years. And then finally, lake whitefish also support one of our largest commercial fisheries that we have here in the Great Lakes. Most of our annual harvest stems from Lakes Michigan and Huron, but lake whitefish commercial fisheries can typically be found in each of the Great Lakes, albeit a little bit less in Lakes Ontario and Lake Erie. So across the Great Lakes, except for Lake Superior in particular, lake whitefish within the last 20 years or so have been experiencing recruitment declines throughout their native range. And this has often been associated to several different factors, such as habitat degradations, overexploitations from our fisheries, disruptions from climate change, reducing available ice cover, essentially for protecting the eggs during the winter season, and then also impacts from invasive species. So back in the 1950s to early 1970s, where sea lamprey invaded the Great Lakes, lake whitefish heavily suffered from population declines because of predation by invasive sea lamprey. In addition to all these factors, there's other factors such as the decline of Diaperia species, which are a high quality prey for lake whitefish. And this is coupled with the invasion of Dreissena mussels beginning somewhere in the early to mid 1990s within each of the Great Lakes. So from this invasion, we've seen that lake whitefish diets have shifted towards more nutritionally inferior species. So instead of feeding on Diaperia and Mysis, for example, which were more abundant in each of the Great Lakes at one point, they're now shifting their diets towards nutritionally inferior prey. For example, alewife, or I should say young of year alewife. They're not piscivorous fish by any means, but they will feed on young of year species, and they'll also be feeding on Dreissena mussels and so forth. The consumption of Dreissena mussels, alewife, and other species that are essentially rich in the enzyme thiominase. Thiominase containing prey can induce thiamine deficiency, and this has been known in several of our other salmonid species we have here in the Great Lakes, like lake trout. But the thiamine deficiency hasn't yet been fully investigated in lake whitefish within the Great Lakes. So this kind of leads me into the goal of my project. And so the objective of my project is to evaluate the potential impact of thiamine deficiency on lake whitefish recruitment. And so to go about this, I'm essentially looking at various dietary components within lake whitefish that may be responsible for leading to thiamine or potential thiamine deficiency and thus affecting natural recruitment. The objectives of my project, a couple of them that I have listed here. One is to determine any correlations between the lake whitefish diets and egg thiamine concentrations. And then the other objective that I have is to assess lake whitefish diets through analyses of fatty acid signatures present in their belly flap tissues. So in the fall of 2024, I ended up collecting lake whitefish from several locations throughout the Great Lakes. If you can kind of see it here in figure 2. It might be a little bit hard to see, but there's approximately 11 sites that I collected data from for this presentation, with plenty more still on the way. So lake whitefish, from the lake whitefish that I collected, I received eggs and belly flap tissues, and this was done through existing surveys of state, federal, tribal, and university surveyors. Thiamine was extracted from the eggs of lake whitefish using and quantified using high performance liquid chromatography, which we conduct here at SUNY Brockport. Lipids were extracted from belly flap tissues, and then fatty acids were transmethylated from those lipids, which is also done here at SUNY Brockport. And this was done through analyses using gas chromatography and mass spectrometry. So some of the results that I am starting to see from my analyses, egg thiamine concentrations don't appear right now to be significantly different among my sampling sites. However, as I mentioned, this is just preliminary data that I've collected so far, and these results may change. We will be collecting, or we'll be conducting another round of fall sampling this fall 2025. Belly flap fatty acid signatures, on the other hand, we are seeing that there is some significant differences between the sampling sites, and then there are also key fatty acids that may be contributing to these spatial differences that we're seeing. For example, 18-1N-9, 16-1N-7, and so forth. The preliminary results that I'm seeing so far, they may suggest that there are some spatial variations in the diet of lake whitefish across the Great Lakes. However, these are just preliminary results. So there is still plenty of more data that I will be collecting for this project. Moving forward, I am also planning to conduct stable isotope analyses from lake whitefish mussel samples. And then we will also be conducting all of these analyses on prey species that I will be collecting beginning this spring. So prey species will be collecting roughly 30 per prey fish species, and then roughly 200 to 300 individuals of, Dreissena mussels, Diperia, mysis, and so forth. Realistically, any prey species that might be consumed by lake whitefish and could contribute to potential thiamine deficiency within the species. I mean, I went through this pretty quick, so I do apologize. I will have plenty more time to talk about any kind of future directions. But right now, I would also like to thank several members that have contributed to this project so far, especially in terms of sample collection and analyses. Big thank you to my advisor, Dr. Renchard, as well as my committee members. Huge thank you goes out to the several organizations that have contributed to collecting lake whitefish and soon to be prey samples for this project. SUNY students, SUNY Brockport students, graduate students, and undergrad students here in the lab at SUNY Brockport that have helped with their analyses, with the lake whitefish analyses so far. And then finally to the Great Lakes Fishery Trust and SUNY Brockport for their financial support in completing this project.
Fantastic. Thank you for your presentation.
Thank you.
Is there anyone in the room who has not had a chance to do a presentation yet? If there is no one in the room who hasn't had a chance to do their presentation yet, then you are welcome to join other breakout rooms if they are still going, or you're welcome to go back to the main room for conference close. As I mentioned earlier, I did put the link in the chat to where these postings will be, where the recordings for these poster sessions will be. We'll need a day or two to get all of them up there. So thank you to all of our presenters today. Please feel free to join another breakout room if they're still going, or head back to the main room. Thanks, everybody.
Thank you.
Thank you.
Thank you very much.